Assalamu alaikum. I am Samra Shahzadi from MS Chemistry Batch 4 and my topic of presentation is nanotechnology based drug delivery system, in which I will discuss about the background, method of drug delivery, targeting of drug, different nanocarrier used in the drug delivery, future of nanomedicine and drug delivery, and the conclusion. So as we all know that nanotechnology has gained a lot of attention since the last few years, and it is expanding rapidly from academic to industry level, as it is one of the fastest growing area of the scientific research and development with the significant advances that are being made, and one of them is drug delivery system that are based on the nanotechnology. So the nanoparticles can be designed to release in a controlled manner that allow the prolonged drug delivery over the period of time. The nanoparticles that are used in drug delivery have size range of 10 to 1000 nanometer, with at least one dimension should be below 100 nanometer. Nano delivery systems are rapidly developing where material in the nanoscale range are employed to serve as a mean of diagnostic tool, means to diagnose various diseases or to deliver the therapeutic agent to a specific target site for the treatment of various diseases in a controlled manner. So nanotechnology offers multiple benefits in treating the long-term human disease by site-specific and target-oriented delivery of the drug. So the large size material basically have some major challenges associated with them, including the in vivo instability, poor bioavailability, poor solubility, probable adverse effect of the drugs. So therefore nanoparticles were introduced. So nanoparticles, on the other hand, nanoparticles can move freely in the human body as compared to the bigger particles, and nanoscale size particles exhibit unique properties like different structural, chemical, mechanical properties, etc. Drugs with the low solubility possess various biopharmaceuticals delivery issues, including the limited bioaccessibility after the intake through the mouth, less diffusion capacity into the outer membrane, and it requires more quantity for the intravenous intake and other unwanted effects. So therefore, these limitations could easily be overcome by the application of nanotechnology in the drug delivery system. The application of nanotechnology in the practice of medicine is known as nanomedicines, and it is also emerging rapidly. So one of the application of nanomedicine is the creation of the nanoscale devices, which is called as nanobots or nanorobots. So they are basically used for improved therapy and diagnostic purposes. These nanobots serve as a vehicle for the delivery of the therapeutic agents and use as a detector for early disease detection and repair of metabolic or genetic defects, deliver them to target tissue more precisely in a controlled manner. So these nanobots even can fight the tumor cells, and they can even invade through the red blood cells to deliver the drug to a specific target. And regarding the use of nanomaterial in a drug delivery, the selection of nanoparticle is based on the physiochemical feature of the drug. Metallic, non-metallic, organic, inorganic, or polymeric different nanostructures, including dendrimer, micelles, and liposomes, can be used as a drug delivery system, which I will discuss in the upcoming slides. Polymeric nanoparticles can be categorized into nanosphere and nanocapsule. So nanosphere are solid core spherical particles in which the drug is embedded within the matrix or adsorbed on the surface, as you can see in the second picture. While the nanocapsule are vesicular system in which the drug is embedded within the central core and which is further surrounded by the polymeric sheet. Method of drug delivery. So there are two ways through which the nanostructure deliver the drug. One is passive and the other is the self-delivery. So in the first one, the drug is incorporated in the inner cavity of the structure, and when the nanostructure material are targeted to a particular site, the intended amount of drug is then released because of low content of the drug, which is embedded in a hydrophobic environment. So on the other hand, in the second method, the drug that are intended for the release are directly combined to the nanostructure carrier for easy delivery. But in this method, the timing is very crucial because maybe the drug will not reach the target site and dissociate from it very quickly, and its bioactivity and its efficiency will be reduced. Targeting of drug. So targeting of drug is another significant aspect that uses the nanomaterial or nanoformulation as a drug delivery system, and it is further classified into active and passive. So in active targeting, different moieties such as antibodies and peptide combine with the drug delivery systems and which help them to anchor to the receptor structures that are present at the target site. While in the passive targeting, the prepared drug carrier circulate through the bloodstream and then reach the target site by the specific affinity or binding that can be affected by different properties like pH, temperature, molecular site and shape. So the main target in the body are the receptors on the cell membrane, lipid component of the cell membrane and the antigen or proteins on the cell membrane. So currently mostly the nanotechnology mediated drug delivery are targeted toward the cancer disease and its cure. There are different nanocarriers that we can use in the drug delivery and I will discuss each of them one by one. So first is the polymeric nanoparticles. So as the name show, they are made up of repeating unit of specific polymers. And some of the non-polymers that are used in the drug delivery are chitosan, polylactic acid, which is also known as PLGA, polylactic acid. So, but PLGA and PLA are the most widely studied, while chitosan research is still under the process. But it has also gained attention due to its biocompatibility and low toxicity. Their size range is 10 to 1000 nanometers. The drug is embedded in the polymer and different surfactants or surface acting agents are used to dissolve the polymer. Organic solvent like acetone, ethylene glycol are also used to dissolve the surfactant and polymer so that the drug is easily entrapped inside the polymer. But they have conditions like the polymer should be biocompatible with the body and tissues. It should be non-toxic and it should be biodegradable, which means that it should break down naturally without causing any harm. They also have advantages like the size is small, so which make the penetration easy through the capillary and the drug accumulate on the target site. It means the targeting easy when we use the polymeric nanoparticles. They are easy to formulate. They don't have stability issues, that why for many pharmaceutical industry use them. They allow the target and they allow the control and targeted drug delivery. For example, in the case of cancer, they even allow the prolonged drug release without any interruption. The next category is lipid based nanoparticle. So basically in this case the lipid is used for the entrapment of the drug. Their size range is 100 to 500 nanometer, and in this type there are two type of formulation we prepared. The first is solid lipid nanoparticles and the other is the nanostructured lipid carrier. The various solid lipid are glyceryl stearate and glyceryl monoxide. So this is the best delivery medium for the bioavailability and enhancement of hydrophobic drug. They also have the same condition like the lipid should be biocompatible and it should be biodegradable. So nanostructured lipid carrier, they are different from the solid lipid nanoparticles. So because in this case both solid lipid and small amount of liquid lipid can be used, and the reason for adding small amount of liquid lipid is that it adds more perfection and more capacity for the drug entrapment. So drug entrapment ability of the nanostructured liquid carrier is more than the solid liquid nanoparticles, and they are also more stable than the solid lipid nanoparticles. The next is the nanosuspension. So nanoparticles suspended in the solvent system called as nanosuspension. The drug powder is dispersed in the solution of the surfactant, and their size range is 200 to 600 nanometer, and they are efficient for the poorly water soluble drug, but they are not very common and they don't have very much literature. The next one is the polymeric micelles. So they are used for the systematic delivery of the water insoluble drugs. Amphiphilic copolymers self-associate to form the micelle in the water, and their size range is less than 100 nanometers. And the advantages are they are very stable, so they provide prolonged stability, and there is no biocompatibility issue for the polymeric micelles, and they accumulate in the tumor cell greatly than the normal cell. And polymeric micelles are reported to be applicable for both drug delivery against the cancer, and they also use for the ocular drug delivery. It means that they can also use to deliver the drug inside the eye, and even they can even reach the posterior ocular tissue. The next one is the liposomes. So they are small artificial spherical shaped molecule. So they are prepared from the natural phospholipid and cholesterol. And the type of liquid, they are basically type of lipid based nanoparticle, but their size is greater, and the size range is 500 nanometer to several micrometers depending upon the type. And they are lipid bilayer structure, as in polymeric micelles the structure was single layer. Both hydrophobic and hydrophilic drugs can incorporate into them. Next advantage is they are also biocompatible and biodegradable, and when drug is introduced in the liposomes, they can reduce the toxicity and also prevent the early degradation. We can also modify the liposome structure to enhance the circulation time in the blood by attaching the polyethylene glycol, and they are also perfect for targeting the cancer tissues. There are different type of liposomes which perform different functions like immunoliposomes, transferosomes, pharmacosomes, ethosomes, and stereosomes. The next is the inorganic nanoparticles. So the inorganic nanoparticles have been widely studied for the potential use in the drug delivery system due to their unique properties such as small size, biocompatibility, and stability. Inorganic nanoparticles such as dendrimer can be used to encapsulate and deliver the drug to a specific target site inside the human body. And one of the inorganic nanoparticles is dendrimers. So dendrimers are highly branched and three-dimensional molecules, and their surface is functionalized easily in a controlled way, and which make these structure excellent candidate as a drug delivery agents, and drug is added inside the core of the dendrimer. So dendrimer can be prepared by two approaches. The first one is called divergent approach, and the second one is called convergent approach. In the first approach, the dendrimer starts its growth from its core and then it grows outward, while in the second case it is opposite. The dendrimer starts its growth from the outside and then grow inward. Dendrimers can be grouped into several kinds according to their functionalization moieties, such as polyamidoamine, polypropylene, and glycodendrimer. And polyamidoamine is the most studied dendrimer, and they are used for the oral drug delivery because it is water soluble and it can pass through the epithelial tissue, boosting their transfer via the paracellular pathway. The drug is basically delivered by the dendrimer following the two different paths. By the in vivo degradation of the drug dendrimer covalent bonding on the basis of the availability of suitable enzymes, or by the favorable environment that could cleave the bond by discharge of the drug due to the changing in the environment like pH and temperature. Next advantages. So one of the main advantage of dendrimer as a drug delivery system is their ability to encapsulate a large amount of drug in the interior or on their surface. So this allows for the control and the sustained release of drug, improving its therapeutic efficiency and reducing its side effects. Another advantage of the dendrimer is their biocompatibility, which is due to their non-toxic and biodegradable nature, which makes them promising platform for the delivery of a wide range of the drug, including the small molecule proteins and nucleic acid. Both hydrophobic and hydrophilic drug can be introduced in them, and they are much more stable than any other formulations. And due to the presence of internal cavity in their globular shape, these macromolecules can help in the targeted drug delivery. But they also have some limitations, like despite their potential benefit, they have different limitations. Like one of them is controlling the size and shape of dendrimers. They can even interact with the immune system and cause toxicity, which limit their use in certain applications. However, more research is needed to fully realize the potential of these nanocarriers and to overcome some of these challenges that are associated with their use, such as toxicity and stability. The next is the protein and the polysaccharide based nanoparticles. Protein based nanoparticles are generally decomposable, can metabolize, and are easy to functionalize for its attachment to a specific drug and other targeting ligands. And they are normally produced from the water soluble proteins like bovine, albumin, and from insoluble like zein and gliadin. And one of the main drawbacks of the polysaccharide is the nanomedicine field is the degradation characteristics at high temperature, which are often required in the industrial process. So besides, most of the polysaccharides are soluble in water, which limits their application in most of the nanomedicine field. However, different techniques such as cross-linking of the polymer can be used to overcome this problem. And there is not very much literature about these, but however they are generated from the biocompatible compounds, so they are excellent candidate for the further development as a drug delivery system. So the last one is the carbon nanotubes. So carbon nanotubes are the one of the nanotechnology most distinctive invention, and their ideal carrier for the drug delivery, as they allow for increased loading capacity in comparison to the conventional nanomaterials. So carbon nanotubes are sp2 hybridized rolled graphene sheet, and the significant characteristics are their smaller size, they have high aspect ratio, They are lightweight, they have excellent conductive properties and excellent tensile strength, which making them efficient. They have vast applications in nanotechnology, nanomedicines as a biosensor, and even bioimaging. Advantages. So the carbon nanotubes can easily penetrate through the cells and deliver the drug directly to the cytoplasm or nucleus. And drug delivery improve the pharmacological and therapeutic profile and effectiveness of the drug and lower the occurrence of off-target. Future of nanomedicine and drug delivery. So the science of nanomedicine is currently among the most fascinating area of the research, and a lot of research in this field in the last 2 decades has already been done. Cancer appear to be the best example of disease where both its diagnosis and therapy have benefited from the nanomedical technologies. So by using various type of nanoparticle for the delivery of the accurate amount of drug to the affected cells such as cancer tumor without disturbing the physiology of the normal cell. The application of nanomedicine and the nanodrug delivery is certainly the trend that will remain to be the future arena of the research and the development for the decade to overcome. And there has been a lot of enthusiasm with the simplistic view of development of nanorobots or nano devices that function in tissue diagnosis and repair mechanism with full extended control mechanism. Conclusion. No doubt nanotechnology has dynamically developed in the recent years, and all the country, whether developed or not, are increasing their investment in their research and development, and nanotechnology advancement have improved the way we view the world, keeping view the immense contribution in the field of medicine. Nanomedicines have already revolutionized the way we discover and administer the drug in the biological system, and thanks to the advances in the nanomedicines, our ability to diagnose the disease have improved a lot. With all the benefits, there are also some setbacks that delay our progress. So nanoparticles are often very reactive and due to their large surface area and surface chemistry. And in addition to the wide application of the nanoparticle in everyday consumable product, they may result in toxic effect due to the repeating exposure to these nanoparticles. So the continuous human exposure to such nanoparticles, either directly or indirectly, they may result in toxic side effects in the future. But unfortunately, there are very few studies that have investigated the biological and environmental impact of the large number of nanoparticles. So as such, there is a need for study and research in the investigation of ecotoxicological effects. So ecotoxicological effects means that the study of the impact of the different substances or harmful chemicals on the population, community, and even on the environment. And another challenge of nanoparticle in biomedicine is their potential use for the immune recognition and the clearance. So the immune system can recognize and eliminate the foreign particles, including the nanoparticles, through a different mechanism such as phagocytosis by the macrophages. So therefore, strategies to minimize the immune recognition and clearance of the nanoparticles such as surface modification and targeting are necessary for effective biomedical application. Furthermore, the stability and reproducibility of the nanoparticle is also a crucial challenge that must be addressed. The physiochemical properties of the nanoparticles such as size, shape, and surface charge can affect their behavior in biological system and the effectiveness of their application. So lastly, the regulatory approval process for the nanoparticles-based technology is still under the development, and it can also be a challenging. So the regulatory agencies require extensive safety and effective testing to ensure the safety of the nanoparticle-based products.